Good morning to all. Today I am going to discuss about the sexual reproduction in Polysiphonia. As you all know that Polysiphonia is polysiphonous in nature, that is, it consisting of a central siphon surrounded by a very pericentral cell except in trichoblast. Okay, so the sexual reproduction is usually an advanced type of oogamy. And most species of Polysiphonia are heterothallic, that is, they having male and female sex organs or sex parts born on separate plants. The male sex organ is known as spermatangium or antheridium, whereas the female sex organ is known as carpon. There are three different types of individuals in Polysiphonia. As I already mentioned in the previous class, in Polysiphonous life cycle is triphasic diplobiontic life cycle. That is consisting of three life phases. That is, one is gametophyte, which is a haploid plant species which bears sex organs. That is, which bears trichoblast, and it is haploid. It is a free living heterothallic plant. Whereas carposporophyte is the starting diploid phase of Polysiphonia. It develops from the zygote which form in the female gametophytic plants, and they develop diploid carpospores. And these carpospores, when liberated, they germinate to form a free living diploid tetrasporophytic plants. That is the third diploid stage of Polysiphonia. In tetrasporophyte, the meiosis happens and it produces tetraspores. Okay, so this is the overall view of what happens in Polysiphonia during sexual reproduction. And in sexual reproduction, as I already mentioned, there are male sex organs which is born on male plants and female sex organs in female plants. On male sex organs, what happens is that you already know that trichoblast is a fertile branch. It is monosiphonous, but what becomes when it starts to develop the male sex organ or female sex organ, what happened is upper portions will become polysiphonous. That is, it began to produce pericentral cells. So the pericentral cells is known as spermatangial. The first pericentral cell formed is known as the first spermatangial mother cells, and each spermatangial mother cells may produce 2 or 4 spermatangia. From the figure you can see 2 or 4 spermatangia, and from 2 or 4 spermatangia they have produced single non-motile, and from each spermatangial mother cell a single non-motile spherical spermatangium is formed. And when mature, the spermatangium liberated in the water by a slit formed in the spermatangial wall, and they carried by water current towards the female sex organs. So this is the formation of male sex organs. Then what happens in female sex organs? It is somewhat more complicated. They are formed in carpogonium, which is developed on carpogonial filament, which in turn developed on female trichoblast. But the carpogonium in overall, it is a flask-shaped structure which is swollen basal portion which bear carpogonium or egg, and a beak, narrow beak, which is known as trichogonium. So what happens during female development? The female trichoblast, it starts as a single only central siphon, but as soon as it becomes fertile, it cut off single pericentral cell, and what happened is, from the lower end of trichoblast, an ensheathen layer of pericentral cell is formed, and one of the pericentral cells of the middle tier, that is in the middle one function as a supporting cell. This supporting cell is very important. The supporting cell, which supporting cells again divides 4 or 5 times to form a short filament. It is known as carpogonial filament. This is which later formed into carpogonium. And the terminal cells of carpogonial filament now differentiates into carpogonium, and the supporting cell will function as supporting. That is, it cut off 2 sterile cells, a basal sterile initial and a lateral sterile initial. These are all important. They, as soon as fertilization happened, they have some role. So both this initial later developed into a short basal sterile filament and a short lateral sterile filament. At this stage, the whole structure is known as procarp. Now it is ready for fertilization, just as start up like a fruit. Now the whole structure is known as procarp. So I just want to summarize what I have said. The female trichoblast, it cut off end sheathing pericentral cells, and the middle tier of pericentral cells function as supporting cell. The supporting cell divides to form 4 or 5 cell short filament known as carpogonial filament. Not only that, the supporting cells also divides to sterile cells, a lateral sterile filament and initial, and a basal sterile initial. The basal sterile initial give rise to 2 to 3 short basal sterile filament and the lateral sterile filament respectively. At this stage, the whole structure is known as procarp, and now it is ready for fertilization. Okay. So during fertilization, what happened is the structure is known as procarp. So now the male spermactangia swims towards the female spermagonium, female carpogonium, and the trichogyne dissolves, and the male nucleus indulges into trichogyne and fuses with the egg or carposporangium, and that begins the start of diploid life cycle of Polysiphonia. The first formed phase is diploid zygote. Then the second important stage in Polysiphonia is the post-fertilization changes. Just like the development of a fruit, some important things happened in Polysiphonia. What happened is the carpogonium undergoes a series of changes, and the whole changes is known as post-fertilization changes. The first and most important change is the formation of auxiliary cell. It is also produced from supporting cell. So supporting cell is very important. It produced carpogonium, it produced basal sterile filament, it produced lateral sterile filament, and now it forms auxiliary cell. And you have to remember that the whole thing happens in the female gametophyte, which is a haploid plant. So what happened is auxiliary cell is also haploid plant. The only diploid one is the zygote in the female plant. Okay. And what happened is the auxiliary cell is produced just beside the carpogonium. So what happened is the auxiliary cell forms a connection, a tubular connection with the carpogonium. So now diploid nucleus divides mitotically, not meiotically, mitotically into two. And what happened is one of the nucleus passes towards the auxiliary cells through the tubular connection, and the haploid cell in the auxiliary cell begins to degenerate. What happened is the result is auxiliary cell also becomes diploid. So there are two cells in the haploid plant. One is the auxiliary cell and the other one is the zygote. Okay. So by this time, what happened is the basal sterile filament, the lateral sterile filaments increase in size. It goes and cover the carpogonium. The pericentral cells just close to the supporting cell produced many vegetative cells also, which ultimately develops into carposporophyte. So what happened is the carpogonium and trichogyne gradually begins to degenerate, because carpogonium gives its nucleus to auxiliary cell. So now carpogonium have no role. So carpogonium begins to degenerate and produced many initials known as gonimoblast initials from the auxiliary cells. So this gonimoblast initials is produced from the auxiliary cells. So what is the result is that the gonimoblast initial is also diploid nucleus. Because the diploid nucleus of auxiliary cells divide mitotically and it gives its nucleus towards the gonimoblast initials. Thus the gonimoblast initial also contain diploid nucleus. So what you have to notice is that only the diploid cells in that female trichoblast sustain, whereas all the haploid cells shows signs of degeneration. So each divides and forms the gonimoblast filament, a short filament known as gonimoblast filament, and the terminal cell of gonimoblast initial swells up and form carposporangium. Okay, just like fruit, it swollen up and forms carposporangium, and each content of each carposporangium from each gonimoblast filaments differentiate into a single diploid carpospore. So by this time, the supporting cell, the auxiliary cell, the carpogonial filament and the sterile filament all fuse to form an irregularly shaped nutritive cells just like placenta, and it is termed as placental cell. The whole structure, the gonimoblast filaments, the carposporangium, the carpospores and the placental cells completely ensheathed by a wall also, and the whole filament look like a urn-shaped structure. You can see from the figure, the whole structure gives the urn-shaped structure which is known as cystocarp or carposporophyte, which is a second the life cycle of Polysiphonia, but it is diploid. All the haploid life cycle becomes degenerate or fused to form placental cell. So, and its sheath is known as pericarp. It has an opening on the upper portion, and the opening is known as osteo. The cystocarp is not free living. You cannot see a diploid cystocarposporophytic plant in Polysiphonia because it is parasitic and it develops inside the haploid female gametophyte. Okay, so this is the second life cycle of Polysiphonia, which is diploid and dependent, parasitic on haploid female gametophyte. So when mature, the carpospores are liberated through the ostiole and they germinate to form the third diploid life cycle in a free living plant, the tetrasporophyte. Okay. Tetrasporophyte. What is the difference is, when you look, the tetrasporophyte and the haploid gametophyte look similar. But when you count the chromosome number, you can see that tetrasporophyte is a diploid plant species, whereas the female gametophyte is a haploid plant. Okay. The tetrasporophyte is a free living plant, but it is an asexual reproductive plant. There is no sexual reproduction occurring in tetrasporophyte. So, in asexual plant is known as tetrasporophyte. Since it is an asexual plant, it bears spermatangium or spermatangia. It does not bear spermatangia, but it bears tetrasporangia. Okay, because why it is tetraspore? Tetraspore is formed because it is a diploid plant. It undergoes meiosis and produced 4 tetraspores, which are arranged tetrahedrally. That is why you get a. This is a three-dimensional structure of tetraspores. Tetrasporangia are produced from the pericentral cells. Not always. You should remember that the central segment doesn't divide to form any reproductive structure or any spores. Only the pericentral cells divide and redivide to form spores and female or male gametes. Okay. So each pericentral cells divide vertically to form an outer cell and inner cell. The outer cell divides and to form 2 or more cover cells, whereas the inner cell have the original role as sporangial mother cell. Sporangial mother cell divide transversely to form a lower stalk cell and an upper tetrasporangial cell. The tetrasporangial cell functions as tetrasporangium. Its diploid nucleus undergoes meiosis to form 4 haploid tetraspores. When mature, they are liberated by the rupturing of the sporangial wall and they germinate to produce the haploid gametophytic plants. Okay. So in Polysiphonia, it is always noticed that out of the 4 tetraspores, 2 germinate into male gametophyte and 2 into female gametophyte. So this is the life cycle of Polysiphonia. It is a triphasic haplodiplontic life cycle having a haploid gametophyte, a diploid carposporophyte and a diploid tetrasporophyte. So hope you understand this section well. Go through the sessions and you can clarify your doubts by asking, by note down your doubts and asking in the next class. Okay, thank you.